Well, thank you. Thanks for the invitation to present this morning. So my presentation then is going to be about cost effectiveness, as Minnie's already introduced. But I thought I might just start with one slide about health economics from a UK perspective at least. And that the key principal question is whether the added value of pharmacogenetic testing, by value I mean gains in health overall, whether they justify the cost. There's an argument that these adverse drug reactions are clearly very costly to manage, and so any test to prevent them would naturally be cost-saving when the cost of testing is increasing or diminishingly small. However, I'll come on to that in a moment because there's a bit more nuanced than that. In the UK, the preferred measure of health outcome is the quality adjusted life year, which is a composite outcome that integrates quality of life over time. So it's a function of both quality of life, health-related quality of life, and life expectancy or survival. From an economics point of view, it's not an accounting exercise. We're not interested just in the cost, but the relation between costs and benefits. And costs we normally take from the payer's perspective, which in the UK is the National Health Service. We then calculate the incremental cost effectiveness ratio, which is the difference in overall costs. It's not just the cost of the testing, but it's the cost of the medicine, the cost of managing the adverse drug reactions, the cost of alternative treatments, etc., divided by the difference in benefits. And if this ratio is below a threshold of about £20,000 to £30,000, then an intervention would be deemed to be cost effective. In the UK, the National Institute for Health and Care Excellence routinely perform cost effectiveness analyses and appraise the evidence in order to introduce new medicines into the NHS. And we often see news headlines about some new cancer therapy that's too expensive, not cost effective and rejected by NICE. And that would be because its incremental cost effectiveness ratio would exceed this range. So that's the context of a health economic evaluation. I'll run through two case studies of single gene tests that we have completed and published. One in relation to carbamazepine with HLA A3101, and one in relation to allopurinol with B5801. So first of all, a little bit of background. Carbamazepine, first-line treatments for epilepsy. Adverse drug reactions occur in about 5% of patients, and they range from macropapular exanthema, which is a sort of a milder rash presentation, which would resolve upon discontinuation of treatment, right through to more severe hypersensitivity reaction, which is associated with mortality, but even worse than that, SJS and 10, which clearly has a very high rate of mortality, with up to 50% of patients with 10 dying over a period of 90 to 180 days. So it's well recognised that B1502 is associated with SJS in certain populations, in Eastern Asian populations, Han Chinese, Thai populations, and other areas of people of those origins. And these individuals should be screened routinely prior to being prescribed carbamazepine in order to avoid the potential for these adverse drug reactions. More recently, HLA A3101 was found to be associated with hypersensitivity reactions to carbamazepine in populations of European Japanese descent. And the presence of this allele increases the risk of cutaneous adverse drug reactions from about 5%, which would be the background rate, up to 26%. And conversely, the absence of the allele would reduce the adverse drug reaction rates from 5% in the untested population down to 3.8%. So there's a differentiation of being able to select for patients at risk of adverse drug reactions through identification of this allele. So we set by to assess whether it would be cost effective to test for HLA A3101 in patients with epilepsy who would be eligible for treatment with carbamazepine from a European population. There's always, in conducting an economic evaluation, we need to model current treatment with the new patient management incorporating testing. And obviously for those who do test positive, there needs to be a change in the management of the epilepsy. And this typically would involve a transfer to another anti-epileptic drug, and lamotrigine would be the drug of choice in this context. And we adopted an NHS perspective for the costing of this economic evaluation. Now, this is rather busy slide, but it represents the model whereby patients enter as newly diagnosed, eligible for carbamazepine, and they would be eligible for testing, and going down this avenue, when they're tested for A3101, they can be tested positive, in which case they would receive lamotrigine automatically, and they could alternatively obviously test negative, in which case they would continue, or they would initiate carbamazepine. And there's a probability of adverse drug reaction occurring with both treatments, and of course a chance of mortality risk, and if they were experiencing an adverse drug reaction, then the treatment of choice would be lamotrigine. Now, the no testing, the standard treatment, which is at the top here, again, there would be a higher risk of adverse drug reaction, the 5%, as I've already mentioned, but patients would be given valproate if they were to experience some of the milder adverse drug reactions. We could stop there, so it's a simple comparison of adverse drug reactions to these anti-epileptic drugs given the results of testing. However, these anti-epileptic drugs have differential impacts on the management of epilepsy, and we were interested in this, so we added another model, which is depicted on the right-hand side here, on the probability of patients being in remission from seizures and having uncontrolled epilepsy, and of course the natural progression over time into death. The results, which would be our base case results related to a clinical trial, the SANAD clinical trial, which represents on average a 38-year-old male who experienced 12 seizures per year. And we found that overall, over a lifetime of treatment based on either testing or no testing, which is current standard of care, there was a very small differential in cost, only £300. But likewise, there was a very small difference in the health gain, 0.023, and that equates to about 8 quality-adjusted days over a lifetime. So while we were avoiding patients from experiencing severe adverse drug reactions, potentially life-threatening adverse drug reactions, the fact that those were particularly rare meant that the overall benefits are very high in the few, but they're diluted across all the patients who needed to be screened. And so this is a typical outcome from an economic evaluation. In this particular case, the cost-effectiveness ratio was £12,800, which is cost-effective. It's below the threshold of £20,000 to £30,000 per QALY. So we recommended that the routine testing would be cost-effective in the context of patients eligible for carbamazepine treatment. However, noting that on average, the health benefits are very small. And that's just a summary of what I've just said there. So the second case study relates to allopurinol, which is the treatment of choice as a urate-lowering therapy in the management of gout. And about 89% of prescribed gout medication is in fact allopurinol, and most patients are able to tolerate it very well. However, it also is associated with rare adverse drug reactions, which are also SJS and 10, but also DRESS is another problematic adverse drug reaction to allopurinol. And the incidence of these is about 7 in 10,000. It's very rare, but obviously very hazardous if a patient were to experience one of these. HLA-B5801 is implicated in allopurinol adverse drug reactions. It's present in about 12 to 18% of Asian populations, but is far less common in European populations, only between 1 to 2%. And so we would expect that testing in populations where the allele is more prevalent would be more cost-effective than testing in populations where the allele is rarer. And the results of the analysis indicated that for the unselected population, whereby nobody's been tested, the incidence of SJS-10 is about 1.6 in 10,000, and DRESS is about 11 in 10,000. But then with in the presence of HLA-B5701, it's increased to 13 per 10,000, and in the absence is 0.1. So the identification through pharmacogenetic testing can separate out those at high risk and those at lower risk. And we set out to conduct an economic evaluation of routine testing for B5801 prior to the initiation of allopurinol for its indication in gout. The routine testing is not currently recommended by the regulatory authorities, and our analysis primarily focused on the Caucasian or European population within an UK NHS setting, though we did conduct some scenario analyses that related to Asian populations where this allele is more prevalent. The model itself it followed in a similar way to the carbamazepine model, in that patients under current standard of care are not genotyped, they are prescribed allopurinol, and there's clearly a probability of an adverse drug event occurring. With a new pharmacogenetic approach, then a positive test would result in a prescription for febuxostat, which is an alternative urate-lowering therapy. Febuxostat is still on patent, and it is far more expensive than allopurinol. The alternative, as I've already mentioned, would be the standard practice, which carries a risk, a small number per 10,000 of population treated. Now, febuxostat itself carries some risk of SJS/TEN, but it is far lower than that of allopurinol. Now, this is the main findings, and apologies again, this is a rather busy slide, but for you, what we did was separate out the costs associated with care and the QALYs associated with care for both the test group and the standard care group. And we divided it by different categories of costs and patient management. So for the first row, the overall management of gout over a patient's lifetime, the expected cost without testing is £5,596, which is identical to the expected cost of the management of the underlying treatment of gout with testing. So there was no differences in patients requiring monitoring, needing to see their general practitioner, being admitted or at least referred to hospital on some occasions. There was a very small difference in QALY resulting from the management of gout being slightly different in these two intervention groups. Obviously, gout is characterised by acute flares, which are very painful and themselves require some anti-inflammatory treatments for management, and there were no differences in either costs or QALYs in those. So the fact that you were tested or not didn't really impact on the likelihood of experiencing flares. In terms of the management of adverse drug reactions, the absolute difference was very low. It's just a pound and a 0.001 QALY. I mean, it's insignificantly small. On the basis that the adverse drug reactions themselves are very rare. If they only occur 1 in 10,000, then on average, you're going to be dividing a relatively high number by 10,000, which results in a very small number, of course. But the main cost drivers relate to the cost of testing, which at the time was about £55 per patient, and the cost of the drug, because febuxostat is more expensive. Now, only a proportion of patients would be prescribed febuxostat, but nevertheless, on average, the difference was about £50, about the equivalent to the price of the test. So overall, if you read the bottom line there, the strategy of testing per patient, on average, would be £100 more expensive, but there would be a health improvement, 0.0023 QALYs, which, again, is a particularly small number. It's just a few additional days of quality just time on average. So just to highlight where the 103 and the 0.0023 come from, well, the £103 difference relates to the cost of the testing and the additional cost of the alternative treatments, the febuxostat. And the difference in the QALYs come from the background management of the gout, nothing to do with the avoidance of the adverse drug reaction. And the incremental cost-effectiveness ratio resulting from these is just short of £45,000 per QALY, meaning that testing in this context would not be cost-effective for the NHS in the UK. And it would require in excess of 11,000 patients to be screened in order to avoid one case of adverse drug reaction. So just to conclude and to summarise that second analysis, that routine testing for B5801 in order to reduce the incidence of cutaneous adverse drug reactions in this population is unlikely to represent a cost-effective use of healthcare resources, with a very high numbers needed to be tested. However, looking at different scenarios, having chronic renal insufficiency puts an increased risk of experiencing adverse drug reactions, and the numbers needed to be tested reduces significantly to just short of 2,700. And likewise, in looking at a different population where allele frequency is much higher, then about 3,000 would be the resulting numbers needed to be tested. So I'll move on to some of the broader considerations from an economics perspective at least, and the first of which relate to factors that influence cost effectiveness, which I've already alluded to in the previous two case studies. And the first one relates to the cost and benefits of the alternative treatment options. And I've mentioned in terms of the treatments for epilepsy, how lamotrigine, sodium valproate, might have differential benefits for the management of epilepsy, and in the case of gout, how febuxostat may be significantly more expensive than allopurinol. And so the cost effectiveness of testing is driven by and large by the benefits and the cost of the alternative course of treatment for those patients who test positive. A second significant driver of cost effectiveness obviously is the rarity of the adverse drug reaction. In the case of abacavir, where hypersensitivity reactions were relatively common, then testing is highly cost effective. But in these cases of SGS and 10, where despite the severity of the adverse drug reaction, the pure fact that they are very rare means that there has to be a very high numbers of patients tested in order to prevent one additional case of adverse drug reaction. There was a publication last year which was a cost effectiveness analysis, this time of B1502 for carbamazepine in patients of Thai and Singaporean populations in the Far East. They conducted the analysis much in the same way as I presented for the carbamazepine, and they found that in this particular population with this allele, they would need to screen 222 patients to avoid one case of SGS and 10. And you would think, well, that's far more effective in some ways than the analysis we found, because the association perhaps being stronger in this context and the allele being more prevalent. But then in order to avoid this one case of adverse drug reaction, you'd had to change a whole load of patients to an alternative, less preferred anti-epileptic drug, meaning that there would be 3 additional patients having uncontrolled epilepsy. So the trade-off is whether one person avoiding SGS-10, how they compare with 3 patients experiencing seizure breakthrough. And you clearly, you might argue, well, the severity of SGS-10 would certainly outweigh 3 additional cases of seizure breakthrough. But then maybe there are some messages here in translating this to the context of gout and other conditions whereby the differences between the probability of an adverse drug reaction and the reduced efficacy will have some implications in terms of the findings. Some of the factors which result in cost effectiveness suggesting very small benefits or marginal cost effectiveness. One might be the fact that the quality adjusted life here, which considers health-related quality of life and time in a given state, doesn't include such things as direct utility to the patient. We've already heard this morning about the fact that somebody has a test result and know how to interpret it, gives them some satisfaction or perhaps some reduced anxiety, well, that in itself would not be incorporated in these analyses, but perhaps it's an important consideration. An age-old problem is the call for more evidence, and people arguing that, well, we need an RCT to support this, and clearly in many contexts it's not feasible to conduct RCTs. And so there is a potential role for modelling exercises which makes explicit some of the assumptions made, rather than perhaps doing, or maybe in addition to doing, some prospective analyses. The other aspect which I want to touch on relates to incidental findings, and this clearly is of relevance when there's a panel of genes that need testing, whereby maybe a test for HLA would be inclusive of another test for a P450 enzyme and so on and so forth. And so testing for one would give additional value through the knowing of the results of others. And obviously, having one additional result would come at zero marginal cost, because the evidence and the results of the test, of the panel test, would be there from time zero. And I'll run through this example, which relates to the previous two case studies. The first being a patient who is eligible for carbamazepine, who instead of having a single gene test for A3101 would have a panel test. And for simplicity, this panel only contains two alleles, one which would be for the carbamazepine, obviously, and the other one for the allopurinol case study that I've already mentioned. Now, prospectively, a request for that test would be cost effective, because I've already demonstrated in the context of carbamazepine, £12,800 per quality, it'd be cost effective to order the test, and you'd get the free results for allopurinol, and it would be cost effective to action on those results at any time in the future when that patient might have developed gout in addition to their epilepsy. So the panel would remain cost effective based on the incidental findings for allopurinol. But consider the example where the patient initially had gout, and perhaps the prescriber ordered a panel test on the basis of wanting to know their B5801 status, but then there'd be the incidental findings in relation to the 3101 for carbamazepine. Well, it wouldn't be cost effective in this context, because the initial test wouldn't be cost effective to order, and the likelihood of the patient over a lifetime developing epilepsy is so small that it would never be cost effective to have the free results to inform any future prescription of carbamazepine. So there's a contradiction here, there's a sequencing order. Now, I know this is a simplified example with only two particular genes on this panel, and if you had a panel of 50, then obviously I'm sure the story would be quite different, because there would be a higher probability that one or more would have implications for future prescribing. But I think there's a note of caution here that just because a panel gives more evidence than a single gene test doesn't necessarily translate to that it would be cost effective to request the results of a panel. I'll just move on to something slightly different, which is an exercise we did a couple of years ago now, which was a discrete choice experiment, which is a method used in economics and other disciplines to identify which attributes are of importance to patients and to neurologists in this case, in terms of whether they wanted a pharmacogenetic test to inform their prescription. So the discrete choice experiment was conducted in 82 people with epilepsy and in 83 neurologists. The neurologist discrete choice experiment posed a series of questions, and this is one example, which is that the cost of testing would be £35. They would get the result of the test within 2 days, and it would have a 2% positive predictive value, a 70% negative predictive value, and they would be, there would be coverage, they'd be reimbursed on the basis of only in the context of serious adverse drug reactions. We also had an attribute here whether the requirements for testing was specified in the British National Formulary. And we presented that scenario to a neurologist and asked him, would you select this test prior to the prescription of carbamazepine, yes or no? And obviously what we did then was vary these numbers, presented 16 different scenarios, get them to tick each and every one of them, and repeated that for 80-odd neurologists. And in that way, you can assess quantitatively how they trade off better test characteristics in terms of positive, negative, predictive value against maybe the cost or the turnaround time, or obviously some of the other factors included there. The second discrete choice experiment was in patients with epilepsy, and we found that they were concerned with very different attributes. The neurologists were more to do with process and test characteristics. The patients wanted to know, what's the chances of being tested, reduces my chance of having breakthrough seizures, reduces my chance of mild or severe adverse drug reactions, etc. And so this is the framework that we arrived at for the discrete choice experiment, presenting two hypothetical medicines for epilepsy with differences in probability of stopping seizures. So that relates to the effectiveness of the treatment, whether they might experience a skin rash, mild adverse drug reaction, or perhaps a more severe life-threatening one, even though at far reduced probability. And again, we would vary these levels and present several different scenarios to patients to complete. And then we would analyse the results. We found that patients were indeed willing to reduce the chance of seizure remission by 0.6 percentage points for a 1% point reduction in skin rash. So they were willing to give up some efficacy to avoid some adverse drug reaction, some mild adverse drug reaction, and even greater efficacy, 3 percentage points for a 1% reduction in memory problems, which is quite a difficult one, whether that's associated with epilepsy or the treatment for epilepsy. And again, a 1.76 percentage point for a very, very small percentage point reduction in the risk of severe adverse drug reaction. So patients were very risk averse. They were willing to give up quite a lot of efficacy to avoid adverse drug reactions. Neurologists, on the other hand, were willing to pay for percentage point increases in the test characteristics. So the more, the higher the negative and predictive value was of the test, then the more that they would be willing to pay for that, or rather perhaps willing for the NHS to pay for it. So that exercise showed that patients and clinicians identified different attributes of importance in relation to pharmacogenetic testing, but had generally consistent preferences. My final section of the presentation relates to the comparison between rare disease management and rare disease prevention, and the coinage that prevention is better than cure. Well, we know that orphan medicinal products are being developed at ever increasing rates. I think about 40% of drugs approved by the FDA last year were orphan drugs. And obviously manufacturers are given incentives for developing orphan drugs, which include 10 years of market exclusivity in the European Union, 7 years in the US. And when treatments for rare conditions are approved, then they are given greater latitude in assessment of cost effectiveness by the reimbursement authorities. And so what I did was to compare the features of a treatment for a rare condition with genotyping to prevent a rare condition from occurring. And the examples given here, afamelanotide, which is a treatment for erythropoietic protoporphyria, which is a skin condition which occurs in the presence of sunlight. And it's exceptionally rare, less than 2 cases per 100,000 population. But the features are that people have a burning sensation and pain upon brief exposure to sunlight. But prolonged exposure leads to erythema and edema, and there are some severe potential complications. It's associated with liver disease, The clinical evidence that led to this drug being approved by the regulatory authorities include a series of randomized controlled trials, which in total included 259 patients. On the right-hand side is my example of B5801 to prevent allopurinol-induced SJS-10, which occurs in 7 patients per 10,000 patients treated. As I've already mentioned, SJS-10 are particularly severe, more severe than these characteristics for the comparator here. And the clinical evidence, we did a systematic review meta-analysis of the evidence, and there were 13 genetic association studies that included 239 cases of SJS-10. So they're comparable clinical evidence. Okay, one's an RCT, one's association studies, but similar numbers at least. Now, if we compare the costs, genotyping is about £55 per patient, whereas this particular drug is £55,000 per patient. So it's 1,000-fold more expensive on a per patient basis. But one of them, obviously, is for the prevention, whereas one is for the treatment. So for the treatment, the gains in health are very high, between 0.6 and 3.3 quality-adjusted life years, which is quite significant. New medicines coming through have a median quality gain of about 0.1. So, this is a significant, on that basis, this represents a very effective treatment. But the quality gain on average for all those genotyped prior to being prescribed allopurinol is 0.0023, so it's very small on an average of the population. Cost effectiveness, £208,000 to £1 million per quality adjusted life year. Now, it's being approved as we speak by NICE, and there'll be a decision made in May, so we don't know whether it'll be approved by the NHS and whether the NHS will consider this to be cost effective. But it has been approved and it is reimbursed in other countries in Europe, in Austria, Germany, Italy, Netherlands and Switzerland. And recall my first slide, I said that the cost effectiveness threshold is £20,000 to £30,000 per quality. But because this is a rare disease, and because appraisal, decision makers allowed some latitude around that, then they quite routinely approve medicines upwards of £200,000 per quality. So there's a tenfold difference in cost, lack of cost effectiveness. By contrast, my earlier analysis suggested that the genotyping in context of gout is about £45,000 per quality, which would not be deemed to be cost effective because it isn't a treatment for a rare condition. So the conclusions are contradictory. One's a treatment for a rare condition, one is to prevent a rare condition. On face value, one is far more cost effective than the other, but I know which one will be approved and which one will not be adopted routinely. So just to summarise these differences, that orphan drugs treat the identifiable few, whereas pharmacogenetic tests identify the few who are at risk. But the numbers needed to be screened for allopurinol exceed 10,000, so you really do need to have, it's associated with a high numbers needed to screen, contrasting with orphan drugs which have a low numbers needed to treat. So orphan drugs have it very expensive on an individual patient basis, whereas genetic tests are inexpensive for individual patients, but expensive for populations. Orphan drugs provide significant health benefits to those who are treated, whereas pharmacogenetic tests benefit only a small proportion of those tested. So just to conclude with a couple of recommendations, that panels, and even extending beyond that to whole genome sequencing, are certainly more likely to be cost effective than a single gene test, but it's not a given. I think there needs to be further work in this to see under what circumstances and which combinations of alleles to include in a panel would make a panel to be cost effective. There's an issue about acceptance of modelling studies versus the need for direct evidence on clinical effectiveness and the role for modelling. I've described one form of modelling, and obviously within patients with reduced renal function, then it's sufficient by regulators to have evidence on pharmacokinetic modelling without the need of a prospective trial, as Munir and I had in a publication a couple of years ago. So I think there needs to be greater awareness that evidence from modelling would probably be sufficiently robust in many contexts. And finally, that we need to resolve some of these inconsistencies relating to the cost and the reimbursement issues for rare disease management, that is, prevention versus treatment. So I'd like to acknowledge my colleague at Bangor, Catherine Plumpton in particular, and obviously with Munir and others at Liverpool for their involvement in many of these studies. Thank you.